Assalamu alaikum. I'm Dr. Noura Mustafa. I'm a lecturer of medical biochemistry. Today we are going to discuss lipid chemistry. These are the items we are going to discuss inshallah. Biological importance of lipids, chemistry of fatty acids, classification of lipids, chemistry and main functions of these types of lipids. Lipids are heterogeneous compounds related to fatty acids. They are different compounds which are related to fatty acids. They are insoluble in water. Insoluble in water, called hydrophobic. Hydro means water, phobic means hate. They hate water, so they are insoluble in water. But they are soluble in nonpolar solvents, as ether, acetone, and chloroform. So they are soluble in nonpolar, but insoluble in water. Our first objective is to identify the biological importance of lipids. Lipids can act as a source of energy because they yield twice the energy produced by the same weight of carbohydrates or proteins, so they serve as energy store. Also, they contain essential fatty acids, which we will discuss later, inshallah. They act as natural solvent for fat-soluble vitamins, which means they are very important for the absorption of fat-soluble vitamin. The lipids are very important for absorption of fat-soluble vitamin, which are vitamin A, D, E, and K. Lipids have very important role in protection and fixation of the internal organs as the kidneys. The kidney has perinephric fat, which is very important for its protection. The myelin sheath of the nerve fibers, which is an envelope around the nerve fiber, contains lipid. This lipid acts as electrical insulator because it prevents the dispersion of the electric impulse outside the nerve fibers. The lipids under the skin serve as thermal insulator. It helps to keep the body temperature warm, for example, in cold weather. And lastly, lipoproteins. Lipoproteins enter in the structure of the cell membrane and mitochondria. So they are very important in cell structure, and also they are very important for lipid transport in the blood. So to sum up the biological importance of lipids, at first they act as a very important source of energy because they yield twice the energy produced by the same weight of carbohydrates or proteins, so they serve as energy store in case of carbohydrate deficiency. They contain essential fatty acids which have very important structure, and also they act as natural solvent for fat-soluble vitamins. They are very important for protection and fixation of internal organs as kidneys. They act as thermal insulator and electrical insulator. Electrical insulator because they enter in the structure of myelin sheath, and thermal insulator due to their presence under the skin. And lastly, lipoproteins, which are very important in cell structure and lipid transport in the blood. And now moving to our second objective, chemistry of fatty acids. The fatty acids are organic acids. They are monocarboxylic organic acids. They usually contain even number of carbon atoms. Usually, the fatty acid is written as CH3, CH2, CH2, CH2 according to the number of carbon atoms, and lastly COOH. So the general formula is written as RCOOH. Fatty acids are numbered starting from the terminal methyl group. So, for example, if this is a fatty acid, it's written as CH3, CH2, CH2, and lastly COOH. This methyl group is numbered as omega 1, and then omega 2, omega 3, omega 4, omega 5, and so on. The fatty acids are classified into saturated and unsaturated fatty acid according to absence or presence of double bond. At first, saturated fatty acids, they have no double bonds. While the unsaturated fatty acid, they either have one or more double bond. If they contain only one double bond, they are called mono-unsaturated. So mono means one double bond. While if they contain more than one double bond, they are called poly. Poly is more than one double bond. The monounsaturated fatty acid, they contain one double bond, only one double bond, so they are called mono. So here, the monounsaturated fatty acid contain only one double bond in its structure. We have only one example here for monounsaturated fatty acid. It's called palmitoleic acid. The palmitoleic acid contains 16 carbons and contain only one double bond at omega 7 carbon. This is the first carbon, which is omega 1, and 5 carbons. So we have now omega 6, and this is the seventh carbon, carbon number 7, omega 7. So here, number 1 and 5 carbons here. So omega 6, we reached now omega 6. This is omega 7. So between omega 7 and omega 8, this is the double bond. So it's called omega 7, which means the double bond is between carbon number 7 and the carbon number 8. So it contains 16 carbons, only one double bond at omega 7. While the polyunsaturated fatty acid contain more than one double bond. So this fatty acid, for example, contain two double bonds. We have three examples for polyunsaturated fatty acids. They are called linoleic, linolenic, and arachidonic. Linoleic, linolenic, arachidonic. The linoleic contain 18 carbons. Linolenic contain 18 carbons, and arachidonic acid contains 20 carbons. So 18, 18, 20. Linoleic, linolenic, arachidonic. 18 carbons, 18 carbons, 20 carbons. They contain two double bonds, three double bonds, four double bonds. 2, 3, 4. Okay? These two bonds, the first one is present at omega 6. So this is omega 1 and 4. So now we reached omega 5. This is omega 6. So the first double bond is at omega 6. Okay? But there are another double bond. So we have here two double bonds. The first one at omega 6. Linolenic contain 18 carbons. They have three double bonds. The first one at omega 3. This is omega 1, omega 2, and this is omega 3. So the first double bond is at omega 3, but the linolenic contains three double bonds. The arachidonic acid contains four double bonds. The first one is at omega 6. So this is omega 1 and 4. We reach it now omega 5. This is omega 6. The first double bond is at omega 6. So we have three examples for polyunsaturated fatty acid, which are called linoleic, linolenic, arachidonic. Linoleic, linolenic, arachidonic. 18, 18, 20. 18, 18, 20. 2, 3, 4 double bonds. 2, 3, 4 double bonds. Omega 6, omega 3, omega 6. Omega 6, omega 3, and omega 6. These polyunsaturated fatty acids are very important. They are called essential fatty acids. These essential fatty acids, or these polyunsaturated fatty acids, are very important for our body, but they cannot be synthesized because the enzymes needed for their synthesis are absent in humans. So they must be taken in diet because they are very important for our body. So again, essential fatty acids, they are polyunsaturated fatty acids which contain more than one double bond. They are essential for our body. They must be taken in diet. So essential means must be taken in diet. This is the meaning of essential. Must be taken in diet. And they are very important for our body, but the enzymes needed for their synthesis are absent in humans. We have three examples: linoleic, linolenic, arachidonic. 18, 18, 20 carbons, 2, 3, 4 double bonds. Of course, we said omega 6, omega 3, omega 6. These polyunsaturated fatty acids, which are called essential fatty acids, are essential for growth. Also, they enter in the synthesis of phospholipids. These phospholipids enter in the structure of the cell membrane, and only the arachidonic acid enter in the structure of prostaglandins. These prostaglandins act as anti-inflammatory. So, essential fatty acids, at first, the definition, they are polyunsaturated fatty acids which contains more than one double bond. They are essential to be taken in diet because the enzymes needed for their synthesis are absent in humans. Examples: linoleic, linolenic, arachidonic, 18, 18, 20, 2, 3, 4 double bond, omega 6, omega 3, omega 6. Their functions, so definition, examples, and importance: they are essential for growth, phospholipid formation, and arachidonic enter in the structure of prostaglandins. And before moving to classification of lipids, we have to study the structure of glycerol. The glycerol is a trihydric alcohol. So it's written as CH2OH, CHOH, and CH2OH. The glycerol can bind with one or more fatty acids. So at first, this is the glycerol, okay, CH2OH, CHOH, CH2OH. It can bind with a fatty acid, which is written as RCOOH, okay? The fatty acid, its general formula, as we said, RCOOH. So it can bind with only one fatty acid and forming ester bond. This compound is called monoacyl glycerol. This is the glycerol here, okay? And this is the fatty acid. So mono means one, acyl means one fatty acid, glycerol. One fatty acid bound with glycerol, monoacyl glycerol. If two fatty acids bind with glycerol, two fatty acids bind with glycerol, the compound is called diacylglycerol. So this is the first fatty acid forming ester bond, and this is the second fatty acid forming another ester bond. Two fatty acids bound with glycerol, so the compound is called diacylglycerol. Di means two, acyl fatty acid, two fatty acids, diacyl, two fatty acids bound with glycerol. If three fatty acids are bound with glycerol, this is the first, the second fatty acid, and third fatty acid. The compound is called triacylglycerol. Triacyl means three fatty acid. Triacyl means three fatty acid bound with glycerol. This compound is called triacylglycerol or triglyceride. Triglyceride. So the lipids can be classified into simple, complex, and derived lipids. Simple lipids are formed of fatty acids and alcohol. Fatty acids and alcohol. For example, fats and oils. The compound lipids are formed of lipid part and non-lipid part. They have several examples, which we will discuss later. Derived lipids are derived from simple and compound. From simple and compound, the compound is also called conjugated. Simple and compound, when they are hydrolyzed, they form derived lipids. They form derived lipids. Simple lipids are esters of fatty acid with alcohol. So this is the fatty acid and this is alcohol. By formation of one molecule of water, we form ester bond. So this is the simple lipid. The simple lipid here, this is triacylglycerol, this is triglyceride. It's an example of simple lipid. This is the glycerol, which is colored in blue, while this structure, this is the first fatty acid, this is the second fatty acid, and this is the third fatty acid. So three fatty acids bound with glycerol form triacylglycerol, and it's a type of simple lipid. So the example of simple lipids is called fats and oils or triglycerides. What's the structure of triglyceride? Tri means three fatty acids together with glycerol. So the example of simple lipids, fats and oils, are also called triglycerides, which are formed of three fatty acids and glycerol. And now moving to the second type of lipids, which are called compound or conjugated lipids. Compound or conjugated lipids is formed of two parts, lipid part and non-lipid part. We have four examples. Phospholipids, it contains lipid part. If the non-lipid part is called phosphoric acid together with nitrogenous base, it's called phospholipid. If the non-lipid part is carbohydrate, it's called glycolipid. If the non-lipid part is called sulfate, so it's sulfolipid. If the non-lipid part is protein, it's called lipoproteins. We will discuss now phospholipids. Phospholipids is a conjugated or compound lipid. So we have lipid component and non-lipid component. The lipid component is also fatty acids and alcohol. So the lipid component is fatty acid and alcohol. Okay. The non-lipid component is phosphoric acid together with the nitrogenous base. The non-lipid component is phosphoric acid together with the nitrogenous base. So the phospholipid is a conjugated lipid. It contains a lipid part, which are fatty acids and alcohol, and non-lipid part, which is phosphoric acid and nitrogenous base. We have three examples for phospholipid, which are called lecithin, cephalins, and sphingomyelins. Lecithin, cephalins, and sphingomyelins. These three phospholipids are very similar in structure because they also contain this general structure: lipid component, which is fatty acid and alcohol, and non-lipid component, which are phosphoric acid and nitrogenous base. But they differ only in the type of nitrogenous base. At first, lecithin. The lecithin, the nitrogenous base of lecithin is called choline. The nitrogenous base of lecithin is called choline. Lecithin has a very important function. At first, it enters in the structure of the cell membrane. Enter in the structure of the cell membrane. The second function, it acts as a lipotropic factor. What is the meaning of lipotropic factor? It's a substance which prevents the accumulation of lipids in the liver. So it prevents a condition called fatty liver. It prevents fatty liver. So it's a very important substance. So it's called lipotropic factor, which means it's a substance that prevents the accumulation of lipids in the liver. So it prevents fatty liver. Also, it acts as lung surfactant. What is the meaning of lung surfactant? It's a substance which line the lung alveoli. It's a substance which line the lung alveoli. So it prevents lung collapse. It prevents lung collapse. It's a substance that line the lung alveoli between the alveoli, okay, lining the alveoli and air inside the alveoli. So it lines the alveoli, between the wall of the alveoli and air. So it prevents lung collapse. Okay, so again, the functions of lecithin. At first, it enters in the structure of the cell membrane, and the second one, it acts as lipotropic factor. It's a substance which, when present in the liver, it prevents the accumulation of lipids in the liver. And so this is very beneficial because it prevents fatty liver. And the third function, it acts as lung surfactant, which means that it's a substance which lines the lung alveoli. It's a substance which lines the lung alveoli, okay, between the wall of the alveoli and air. And this prevents the lung collapse. If lecithin is deficient, for example, in premature infants, the lung alveoli don't secrete sufficient amount of lecithin. It leads to lung collapse. So these alveoli are normal, while these alveoli are collapsed. They are collapsed due to deficiency of lecithin, which is not secreted by the lung alveoli in premature infants. This condition is called respiratory distress syndrome. So there is distress in respiration due to deficiency of lecithin, which line the lung alveoli and prevent lung collapse. So if this lecithin is deficient, it leads to lung collapse. So the first phospholipid is called lecithin. The nitrogenous base in lecithin is called choline. Okay, its function, the lecithin has very important role. It enters in the structure of the cell membrane. Number two, it acts as lipotropic factor, preventing accumulation of lipids in the liver. And lastly, it acts as lung surfactant, which means that it lines the lung alveoli and prevents lung collapse. If this lecithin is deficient, for example, in premature infants, it leads to respiratory distress syndrome. Cephalins, the nitrogenous base of cephalins are called ethanolamine or serine. So in lecithin, it's called choline. In cephalins, they are called cephalins or cephalins, okay? In cephalins or cephalins, they are called ethanolamine or serine. The cephalins have very important role in blood coagulation because they enter in the structure of what's called thromboplastin. The thromboplastin is essential for blood clotting. So the cephalins enter in the structure of thromboplastin, which has very important role in blood coagulation. So the functions of cephalins are blood coagulation, very important in blood coagulation because they enter in the structure of thromboplastin. And the last example of phospholipid is called sphingomyelins. The sphingomyelins have two nitrogenous bases, sphingosine base and choline base. This choline was also present in lecithin, okay? Sphingomyelins contain sphingosine base and choline base. As its name implies, sphingomyelins is present in the myelin sheath. So it's present, very important, it's abundant in the nervous system in the myelin sheath to act as electrical insulator. So these were phospholipids. They have lipid part and non-lipid part. Lipid part, fatty acid and alcohol, while the non-lipid part is phosphoric acid and nitrogenous base. We have three examples: lecithin, cephalins, and sphingomyelins. All of them contain nitrogenous base, which differ according to the type of phospholipid. Lecithin contain choline, cephalin contain ethanolamine or serine, and sphingomyelins contain sphingosine together with choline. Functions of lecithin: enter in the structure of cell membrane, lipotropic factor, and lung surfactant. Okay, and its deficiency lead to respiratory distress syndrome. Cephalins are important in blood coagulation, while sphingomyelins, they are abundant in the myelin sheath in nervous system, so they are very important as electrical insulator. Now moving to the second type of conjugated or complex lipid, they are called glycolipids. Glycolipids, which means the lipid part is also fatty acid and alcohol, while the non-lipid part is called carbohydrate. Also, they contain sphingosine base. The example of glycolipids are called cerebrozytes. Cerebro means they are present mainly in brains and nerves. They are present in the nervous tissues, in brain and nerves. They act as electrical insulator. And now moving to the last type of lipids, which is called lipoproteins. Lipoproteins mean there is a protein part and lipid part. Protein part and lipid part. The lipids are triglycerides, cholesterol, phospholipids, and cholesterol ester. Okay? Triglycerides, phospholipids, cholesterol or cholesterol ester. Lipoproteins have very important functions. They enter in the structure of cell membrane and they are very important for lipid transport in the blood. As we mentioned at the beginning of our lecture, that lipids are insoluble in water. So how are they transported in the blood? They bind to protein to make them soluble in the blood. Okay, so they bind to protein to make lipoproteins water soluble, so they are transported in the blood. Okay, so lipoproteins are very important for two functions: structure of the cell membrane, and the second, lipid transport in the blood. We have four types of lipoproteins: chylomicrons, chylomicrons, VLDL, VLDL, very low density lipoprotein, which is abbreviated VLDL, very low density lipoprotein, low density lipoprotein, abbreviated LDL, and high density lipoprotein, which is abbreviated HDL. Again, we have four types: chylomicrons, very low density lipoprotein, VLDL, LDL, and HDL. So we have four types of lipoproteins: chylomicrons, VLDL, LDL, and HDL. Okay, all of them contain proteins and lipids. The numbers colored in red show the percentage of total lipids in these lipoproteins. Of course, these numbers show that the chylomicrons have the highest percentage of total lipids, while the numbers colored in blue shows the percentage of total proteins in these lipoproteins, and this shows that the highest percentage of total protein is present in HDL. So chylomicrons have the highest percentage of total lipids, while HDL contains the highest percentage of proteins. The lipoproteins can be separated by either two methods: ultracentrifugation and electrophoresis. What's the meaning of ultracentrifugation? It means centrifugation at very high speed. At very high speed. So the lipoproteins are separated according to their degree of flotation. The chylomicron, of course, as we said, contains the highest content of lipid. So the first is chylomicrons, and then VLDL, LDL, HDL. So the first is chylomicron because it contains the highest percentage of total lipids. This is the first one. Ultracentrifugation means centrifugation at very high speed. They are separated according to degree of flotation. So as chylomicrons contain the highest percentage of total lipids, they are floated first. They are floated first because they contain the highest percentage of total lipids. The second method is called electrophoresis. Phoresis means separation. Separation using electric field. Separation using electric field. So it leads to migration of lipoproteins in electric field and their separation according to two factors: amount of charge and molecular weight. Their weight and amount of charge. Okay, so the order is chylomicron at first, and then LDL, then VLDL, and lastly HDL. The first one is chylomicron, and then LDL, VLDL, and lastly HDL. The LDL is called beta lipoproteins. Why is it called beta? Because it migrates at the same site of beta globulin. What's beta globulin? It's a type of plasma protein. When it's separated by electrophoresis, it migrates at this position. So LDL is called beta lipoprotein because it migrates at the same position of beta globulin plasma protein. While VLDL migrates before beta, so it's called pre-beta. And lastly, HDL is called alpha. So again, the order is chylomicron, LDL, VLDL, and lastly HDL. So we have two methods for separation of lipoproteins: ultracentrifugation, which means centrifugation or rotation at very high speed, separating the four lipoproteins according to their degree of flotation. Okay? Since the chylomicrons contain the highest percentage of total lipids, so it floats number one. Okay? Takes the order number one, and then the LDL, LDL, and HDL. Okay? While the second method is electrophoresis, which is separation of the lipoproteins by migration in electric field. They are separated according to molecular weight and charge. The molecular weight, the chylomicrons have the largest molecular weight. So it almost doesn't migrate. It remains at the same site, okay? While the HDL migrates rapidly, okay? Migrates rapidly at the site of alpha globulin. So it's called alpha lipoprotein. So HDL is called alpha lipoprotein. VLDL is called pre-beta, pre-beta, okay, before beta. So it's called pre-beta. Low is called beta. So again, to memorize, this is called VL, two words, okay? It's called pre-beta, two words. While low L, this is one word, it's called beta. So very low is the same as pre-beta, while low is the same as beta. So these are the four types of lipoproteins, which are chylomicrons, VLDL, LDL, and HDL. The chylomicrons are synthesized in the small intestine, and they have the largest size of all lipoproteins. Of course, we mentioned that they are formed of protein part and lipid part. So all of them contain proteins, and all of them contains phospholipids, cholesterol, and triglycerides. All of them contain protein, phospholipid, cholesterol, and triglyceride, but the percentage differ among them. So the chylomicron mainly contains triglycerides because its function is to transport exogenous triglycerides from the small intestine to tissues. So it contains mainly triglycerides, okay? So it transports exogenous means from diet, okay? Exogenous triglycerides from the small intestine to the tissues. While the VLDL is synthesized in the liver, large size, it contains also triglycerides mainly. It also contains the others, cholesterol, phospholipids, and proteins, but mainly triglycerides because it transports endogenous triglycerides. While chylomicrons transport exogenous, VLDL transport endogenous. What is the meaning of endogenous? It's present inside the body in the liver. It transports it from the, it's synthesized in the liver, so it transports the endogenous triglyceride from the liver to tissues. While the chylomicron is synthesized in the small intestine and contain triglycerides, so it transports exogenous triglycerides from small intestine to tissues. Here from the liver to tissues, okay? LDL is synthesized in the blood. It has small size. It contains mainly cholesterol and proteins, mainly cholesterol. So it transports cholesterol from liver to tissues, from liver to tissues. And lastly, HDL is synthesized mainly in the liver. It has very small size. It contains mainly proteins and phospholipids together. Proteins and phospholipids. It transports cholesterol from tissues to the liver. From tissues to the liver. This is called reverse cholesterol transport. Reverse cholesterol transport. What is the meaning of reverse? Reverse the way, okay? Reverse cholesterol transport means it's reversal to low density lipoprotein because it transports here from liver to tissues, while here it transports from tissues to liver. So this is called reverse cholesterol transport, which means that it transports cholesterol from tissues to liver in order to be eliminated, while the LDL transports cholesterol from liver to tissues. So again, the chylomicrons are synthesized in the small intestine. It contains mainly triglycerides, so it transports exogenous triglycerides from the small intestine to tissues. The LDL also contains mainly triglycerides and synthesized in the liver, so it transports endogenous triglycerides from liver to tissues. So these two, chylomicrons and VLDL, transport triglycerides. The chylomicron exogenous and the VLDL endogenous. while the LDL and HDL both transport cholesterol. This LDL transports cholesterol from liver to tissues, while HDL from tissues to liver, so it's called reverse cholesterol transport. It transports the cholesterol from tissues to liver in order to be eliminated. Okay? Of course, we mentioned the rate of flotation. This is the highest flotation because it contains mainly or the highest percentage of total lipids, and then high, low, and lowest. The electrophoresis, these chylomicrons don't migrate at all because it has large molecular weight. The VLDL, as we mentioned, very low VL towards, so it's called pre-beta, before beta, pre-beta. LDL migrate with beta, so it's called beta, L beta, and lastly HDL is called alpha, alpha globulin HDL. So to summarize, we have 4 types of lipoproteins, chylomicrons and VLDL. Chylomicrons are synthesized in the small intestine, VLDL synthesized in the liver. Both of them contain mainly triglycerides. The chylomicrons transport triglycerides, exogenous triglycerides, from small intestine to tissues, while the VLDL contain mainly triglycerides. It transports endogenous triglycerides from the liver to tissues. LDL and HDL, both of them transport cholesterol. The LDL transports cholesterol from liver to tissues, while HDL transports cholesterol from tissues to liver, and this is called reverse cholesterol transport in order to be eliminated. So HDL is very important and is very beneficial because it helps the cholesterol to be eliminated in the liver. So it's inversely related to the incidence of coronary atherosclerosis. So it's very beneficial. It's inversely related to the incidence of coronary atherosclerosis because it transports cholesterol to the liver in order to be eliminated. So conjugated lipids contain lipid part and non-lipid part. We have discussed phospholipids, glycolipids, and lipoproteins. Phospholipids as lecithin, cephalins, and sphingomyelins, which contain phosphoric acid and nitrogenous base. Glycolipids contain also nitrogenous base as sphingosine and contain carbohydrate part. They are present in brain and nervous tissue. While lastly, lipoproteins are formed of lipid part together with protein part, chylomicrons, VLDL, LDL, and HDL, and we have differentiated the 4 types of these lipoproteins. Lastly, derived lipids. The derived lipids are derived from simple and conjugated lipids by hydrolysis. For example, fatty acids and glycerol. They include substances related to lipids. We have here very important examples: cholesterol, steroid hormones, vitamin D, bile acids and salts. All of them are called derived lipids. All of them contain a nucleus called the steroid nucleus. So they are called steroids. So again, the derived lipids are derived from simple and compound lipids by hydrolysis. For example, fatty acids, glycerol, and steroids. These are 3 examples of derived lipids: fatty acids, glycerol, and steroids. The steroids are compounds related to lipids. For example, cholesterol, steroid hormones, vitamin D, bile acids, and salts. All of them contain steroid nucleus. So to summarize, we have discussed the biological importance of lipids, chemistry of fatty acids, and classification of lipids. At first, biological importance of lipids. Lipids act as a very important source of energy and act as energy store. They contain essential fatty acids, which have very important functions, natural solvents for fat-soluble vitamins, protection and fixation of internal organs, electrical and thermal insulator, and lastly, lipoproteins. The fatty acids, then moving to chemistry of fatty acids, they are monocarboxylic organic acids. They are numbered starting from the terminal methyl group or omega carbon. They are classified into saturated and unsaturated fatty acids according to absence or presence of double bond. The saturated have no double bonds at all, while the unsaturated have 1 or more double bonds. Monounsaturated contain only 1 double bond. And we have one example by metolake, which contains 16 carbon and 1 double bond at omega 7. While the polyunsaturated fatty acids, they contain more than 1 double bond. We have 3 examples: linoleic, linolenic, and arachidonic. which are called essential fatty acids. Essential fatty acids, their definition is polyunsaturated fatty acid which contain more than one double bond, must be taken in diet because the enzymes needed for their synthesis are absent. So this is the definition. Examples: linoleic, linolenic, arachidonic, 18, 18, 20, 2, 3, 4 double bond, omega-6, omega-3, omega-6. Definition, examples, and function: essential for growth, phospholipids formation, and arachidonic enter in the structure of prostaglandins which act as anti-inflammatory. Moving to classification of lipids, we have simple, complex, and derived. We have three types of lipids: simple, complex, and derived. Simple lipids, for example, fat or oil, fat and oil, or triglyceride, fat and oils, or triglycerides, which are formed of three fatty acids and glycerol. Then moving to compound lipids, is formed of lipid part and non-lipid part. The first is phospholipids, which contain lipid part, fatty acid, and alcohol. The non-lipid part is phosphoric acid and nitrogenous base. We have three examples: lecithin, cephalin, and sphingomyelin differ in the type of nitrogenous base. Lecithin, the nitrogenous base is choline. Cephalin is ethanolamine or serine, and lastly sphingomyelin is sphingosine and choline. The functions of lecithin is structure of the cell membrane, lipotropic factor preventing accumulation of lipids in the liver, and lung surfactant. The deficiency of lecithin leads to respiratory distress syndrome, which occurs in premature infants due to deficiency of lecithin, which leads to lung collapse. The cephalins are very important in blood coagulation, and lastly sphingomyelin are abundant in the nervous system in myelin sheath. Glycolipids, for example, cerebrosides, they are present in the brain and nerves, and they act as electrical insulator. They contain lipid part and non-lipid part. The non-lipid part is carbohydrate. The lipoproteins are important in cell structure, in the structure of the cell membrane and lipid transport in the blood. We have four types of lipoproteins: the chylomicrons and VLDL. Both of them transport triglycerides. Chylomicron transport exogenous triglycerides from small intestine to tissues. The VLDL transport endogenous triglycerides from the liver to tissues, while LDL and HDL, both of them transport cholesterol. LDL from liver to tissues and HDL from tissues to liver in order to be eliminated. So it's called reverse cholesterol transport. Of course, according to the flotation, the chylomicron have the highest flotation, and then VLDL, LDL, and lastly HDL. While electrophoresis, the chylomicron don't migrate. VL pre-beta, L is called beta, and lastly HDL is called alpha. The HDL is beneficial. Why? Because it helps the cholesterol to be eliminated from the liver. It transports cholesterol from tissues to liver in order to be eliminated. And lastly, derived lipid like fatty acids, glycerol, and compounds related to lipids as steroids, for example, cholesterol, vitamin D, bile acids, and salts. Okay? Thank you.